Thank you for your time this evening. So I will have I've spoken at a couple of these events before, and some of you will know the Silence Therapeutics story. So for those of you, I'm going to give you a little bit of an update on recent developments, and most notably our decision to take a direct listing on Nasdaq. I'm also going to talk about our technology and our lead programs, and I'm going to spend most of the time in terms of the lead programs on a really interesting program for cardiovascular disease called SLN 360. So for those of you who aren't familiar with Silence, we've been listed on AIM for a number of years. We're pioneers in the field of silencing RNA, and I'm going to spend a few minutes on the basic biology here, because I think it's important that people understand the technology. We have a corporate headquarters here in London, but we have research facilities in Berlin, and the scientific heritage of the company is in Berlin. We have about 50 or so employees today, but we are recruiting very rapidly, and that will increase significantly over the course of the next few months. As I said, listed on AIM, about $450 million market cap. This time last year, we had a $50 million market cap, and really, I think the big step up in valuation over that time has been an external validation of our platform. We've always believed in the strength of our science, but over the last 9 months, we've done 3 significant business development deals with Mallinckrodt last summer, with Takeda in January of this year, and with AstraZeneca in March of this year. And really, I think all of those collaborations have really put us on the map in terms of validating the technology and really kind of signposting the strength of it. We have a really strong financial position. As of the end of March, we had £90 million on a pro forma basis. That takes us into 2023, so cashed beyond 3 years. And importantly, that gives us enough time to read out 2 phase 1b clinical trials for our 2 lead programs, and I'll tell you a little bit more about that in a moment. So a few minutes just on the science and what we're trying to do with our technology. And as many of you will appreciate, given you've all shown the interest in coming tonight to hear biotech companies, you'll probably know a lot of this. But you've got your genetic blueprint, and that is the blueprint for you, what makes you, and that resides in your cells, each of your cells. And the way that kind of is given effect or is brought to life is through being made into proteins. So the DNA in the nucleus gets made into proteins, and proteins do all of the work. The hemoglobin in your blood carries the oxygen around your blood. Enzymes are made of protein. Now, in disease, you often get dysfunctional proteins, so proteins that are made in the wrong way or that for other reasons cause disease. In between DNA and protein, there's a middleman called messenger RNA. And so this is really just a way of capturing the message in the DNA and taking it outside of the nucleus in the cell and to be made into a protein. So this is where Silence's technology comes in. So what we've developed is a technology called Rna Interference or silencing RNA, whereby we very precisely can knock down the target that we're going after. So if you have a faulty messenger RNA, we design a drug that very precisely binds against it. When that drug is administered to the patient, what happens is that middleman gets kind of pushed out of the way, and the faulty protein is no longer made or made at much reduced levels. So that's our technology. Now, that technology in a test tube is very, very reliable and very reproducible, and has been for about 20 years. What was always difficult for companies with our technology was getting it into cell types, so injecting it into patients and making sure that it got into the cell type that you were trying to get to. We've cracked that now. Since 2016, we have a technology called GalNAc siRNA. GalNAc is a simple sugar, but when we put it on the end of our drug, what happens is when you inject it into the patients, the drug is very precisely taken up by one particular cell type, and really only one cell type. So we now have a mechanism to take our technology, decide the DNA sequence, messenger RNA that we're trying to go after, and then put this GalNAc on it and really precisely target it to both the sequence itself through the sequence complementarity and the liver cell through the use of GalNAc. This technology is both robust and durable. So on the slide, you can see on the right-hand side the robustness of the technology. So the two examples shown as a control, you can see the gray bar, which is just a given level of messenger RNA for a target. When you administer the drug, you see that very rapidly you get almost none of the target left. So let's say 95% of it is reduced. That's the durability. That's the robustness of the approach. And then the durability is, well, look at the slide, the chart on the bottom right. You can see that when you administer the drug, the effect can last, in this instance, up to 3 months. And this is a technology that reproducibly and really for almost every target we've looked at, you can get at least a monthly dosing effect, if not every 2 months or every 3 months or even every 6 months, as one of our competitors using similar technology has shown that they can take it out to 6 months with the target that they're going after. So this technology is Silence is a platform to product play. So hopefully you've got a bit of a sense for what that platform is, the underlying technology that we possess. That is valuable in and of itself, but the real value is the fact that you can take that platform and you can turn it into an almost unlimited number of drugs. So we have a couple of drugs nearing clinical development. We have several drugs in earlier stage work, and we have the ability to really translate that platform into numerous products, and we will do that over the coming years. So real, when you think of the value of Silence, you've got to the value of the individual drugs we're developing is an important part of it, but the platform is a huge additional value here. So turning back to the collaborations that we've done over the last 9 months, and I'll do it in chronological order. So last summer we did a deal with Mallinckrodt Pharmaceuticals. This is in the complement space. Complement is part of the innate immune response, and Mallinckrodt took an option over 3 targets in this disease area. So one of those targets we already had in development. We call it SLN 500, and that's progressing well. It is just about to enter IND enabling studies or preclinical studies, and will enter the clinic in late 2021, early 2022. We received 25 million in upfront payments, 5 million of that for equity. And the total deal value is over 2.1 billion in potential milestones if all 3 assets get developed fully. That was a really important deal for Silence, and it really put us on the map. And what it showed, and what's very important, is that it showed other big pharmaceutical players that one of their peers, Mallinckrodt, is more of a specialty player, but they've chosen to invest in Silence, and so the interest levels really picked up. Then in January we signed a smaller deal with Takeda, which we're confident will turn into something bigger. This is a technology evaluation agreement. So Takeda have a novel target. It is undisclosed in an undisclosed indication. So I'm really teasing you. There's not a lot I can say about it, but it is a part of that confidentiality is the Takeda conviction around this target. So we're working together on that over the next 6 to 9 months or so, and then that'll be in a position where Takeda will decide if they want to take it forward or not. Clearly, if they take it forward, then a full license agreement would need to be negotiated. And then really the cherry on top was a deal we did with AstraZeneca on the 25th of March this year. This was really right in the middle of the coronavirus panic from a capital markets perspective. Most biotechs were down 10, 20, 30, 40% for the year at this point, but we managed to get this deal across the line, and thankfully AstraZeneca took the long view. This is a multi-target deal, so this is up to 10 targets. These are all novel targets. This is not any of our existing pipeline. This is not anything we have in early discovery phase. In fact, it's not even anything we have in our in silico portfolio, as it were. Anything kind of any targets we were working up. So these are all novel targets, and importantly, it's both hepatic and extrahepatic. And what do I mean by that? Well, the GalNAc technology that we possess and we are experts in allows us to get into hepatocyte cells in the liver. We are working on ways of getting into other cell types, but AstraZeneca have already got a toolkit of ways and technologies of bringing oligonucleotides, so our drugs, into other tissue types. So we're going to work together to take our expertise in oligonucleotide drug development and their expertise in drug delivery, and use that to target novel extrahepatic indications, so respiratory, cardio, metabolic, etc. Importantly, Silence has an option to negotiate to co-develop up to 2 of these programs. And I think what we're trying to show there is really our intention to be a serious drug developer. We are very rapidly building out our capabilities in that regard, and investing heavily, and we intend to develop that further over the next coming years. Now, this was an $80 million deal in upfront payments, up to $400 million per target. And so that's 10 times 400, $4 billion in total. And the first additional payment comes upon candidate nomination. So when AstraZeneca decide that we've done enough work that the target is sufficient to take into preclinical development, at that point they will pay us a $10 million option fee for each target they take forward, and we expect the first payment to be made in around about 2 years or so. I'm going to just briefly touch on some existing out licenses, because for long-time followers of Silence, people sometimes ask about them. We do have a small royalty stream from Alnylam, but it really is hundreds of thousands a year over the next few years. It's not a material amount. And we have out-licensed our technology to Quark, a private siRNA drug developer who are in phase 3 trials. Quark is a private company, and we're not privy to really any information on how those trials are progressing. But I think we've really moved on from these out licenses and shown that the AstraZeneca license in particular is of a different order of magnitude. So this is our pipeline. We don't show really any of the early discovery assets that we've got. As I said, we have multiple assets in discovery phase, but we like to just really disclose them when we're confident that we've really got a strong lead candidate and ready to open it up to the world. We don't feel that opening that up any earlier really adds any value. The 2 assets at the top and middle are SLN 360 for cardiovascular disease with high Lp(a), and SLN 124 for beta thalassemia and myelodysplastic syndrome, iron overload disorders. I'm just going to spend a few minutes on SLN 360, and I'll come back to SLN 124 for another evening. But they are both on the cusp of clinical development. And happy to take questions on SLN 124, of course. But really, our excitement is on SLN 360, and I'm going to spend the remaining time on that. So this is a cardiovascular disease target. The target has been known about for many years. It was discovered in 1963, but it's really over the last 10-15 years or so, it's came to be appreciated as a real genuine risk factor for cardiovascular disease. And really a lot of the data that started that movement was epidemiology data coming out of Denmark, where they could look at 250,000 people, and they could say, well, what was the risk of these people having a heart attack, and then strip back all the other factors. So strip out everything else that might be causing a heart attack, and what was left was a very, very clear correlation between high Lp(a) levels and cardiovascular disease. And that's really kind of, the excitement around that is now palpable in cardiovascular specialists around the world. It is an independent genetic risk factor. So it really doesn't matter if you're, fat or thin, exercise, don't exercise. The number that you're given, the Lp(a) number you have, that you're born with, is the same number you live your life with. And so these are the people, the guy or girl who drops dead in a marathon, who's, for all intents and purposes, a very fit individual. They're the types of people that you then realize had extremely high Lp(a) levels. So it's this silent killer that can go with people throughout life. It's hugely prevalent. It's hugely prevalent. So about 10% of the population have dangerously high levels of Lp(a). And that's shown on the right-hand side there. You've got the distribution of Lp(a) in the population. So a lot of us, and most of us, have got very low levels of Lp(a). Now that's a good thing, because remember, what we're doing with our technology is we're knocking down, we're reducing the levels of Lp(a). So if you do that and you get low levels of Lp(a), well, that's okay, because most of us have low levels of Lp(a), and we're perfectly healthy for it. This just shows you some of the increased risk of cardiovascular events with Lp(a). I'm just going to show you some of the data that we've generated, and this is in late stage preclinical development. These are animal models using non-human primates, and we've been able to show that we can achieve a greater than 90% knockdown lasting beyond 2 months. So we think this is a really, really exciting program. Strong knockdown with our molecule, huge prevalence, huge unmet need. And remember, these patients are asymptomatic. They don't necessarily know they're at risk. So what we're trying to do is develop a drug that's very easy to administer. You go to your GP or even do it at home. You take an injection in your abdomen, and then that's it. You go away and come back, 2, 3, 4 months later, and take another injection. And for all of that period, your risk of cardiovascular disease has been significantly reduced. Silence is not the leader here. I think if we were the first in line here with this target, we wouldn't be worth 450 million. We'd be worth a hell of a lot more. We are third in a race to develop a Lp(a) molecule. Novartis is in the lead, and they have just started a phase 3 trial. And one of our competitors, Arrowhead, has a siRNA molecule just about to start a phase 2 trial in partnership with Amgen. But we believe, based on the preclinical data that we've got, that our molecule is certainly competitive with that Arrowhead molecule. And I would just, urge you to remember the world of the statins, when there was, multiple statins developed by big pharma players. And actually, the biggest statin was the one that was launched 6th or 7th. So when you've got a huge market, which this is, surely is. You don't necessarily have to be first, and actually we believe being first is somewhat of a disadvantage, in that you have to do more and more clinical studies in order to get your drug approved. I'm not going to talk about 124, but I'll just end and summarize there. So we think a really exciting technology. Our technology is as good as anyone in the world at this. We've shown that over the last 9 months with 3 business development deals. So gone from 50 million market cap to $450 million market cap. But we're really only just getting started. So later this year, we're going to have 2 programs enter the clinic. They will proceed through phase 1b studies over the course of 2021. And there will be many more programs to follow. a really strong management team. We are recruiting for a CEO. That process is going well, but we're not quite there just yet. Strong cash position, taking us out beyond 3 years. And just to address an obvious question, we also announced recently that we're pursuing a direct listing on Nasdaq. So over the next few months, we will list our shares on Nasdaq and give people the opportunity to buy American depository shares as well as ordinary shares on AIM. The idea there was really to put us in the same pool as our competitors, who are significantly more highly valued than we are, and we think, you know, sitting alongside them and having some of that visibility will help. And really, you know, a lot of people go to Nasdaq to raise money. We don't need to raise money at this point, but clearly having a direct listing puts us in a position to be on Nasdaq should we choose to raise money in future. Thank you all for your time, and I look forward to any questions.
I remember the last time you presented for us at an event, it felt like you were really kind of on the cusp of something, and it's always really great to see that being kind of validated and the kind of evaluation coming through accordingly. Had a few questions about capacity. So you've achieved those 3 deals over the last 9 months.
Yeah.
And I think someone's put here, you've got 13 possible targets you might have mentioned. How realistically, or your current capacity, can you talk about what that is, and then how easy or difficult it is to scale up if you need to?
Yeah, it's a good question, and clearly we're in a very different position from, you know, where we were, you know, 12 months ago. With, you know, our platform, our pipeline has effectively quadrupled over that period. A couple of things to say. So you know, we've been sensible and smart about it in terms of the partnerships that we've signed have time criteria for the number of targets that the partner can put in at any one time. So for AstraZeneca, for example, we envisage 5 targets in the first 3 years, but no more than that. So the next 5 would come beyond that. So similarly for Mallinckrodt, you know, they couldn't exercise all 3 of those targets at once last summer. So we manage it a little bit that way. But clearly, you know, we're going to need a lot more staff and you know, to build out the organization, and we're right in the middle of that, actually. We're, you know, it's amazing. There seems to be people joining, you know, almost on a daily basis. So, you know, I think by the end of this year, you'll probably see about 80 heads or so at Silence, maybe even more, which is a great position to be in. And even, you know, with all the you know, that cash guidance that I gave, you know, that clearly takes into account all of that build up in organizational capability and hiring, and even with that, we're still cashed out beyond 3 years.
And the process of hiring in the current environment, and getting the right skill set, but also having the ability to start work, is that challenging at the moment?
It's been so frankly speaking, it's been a lot easier than we thought it was going to be. We've had great success in hiring over the last couple of months, and you know, we've had several people join us who haven't met any of their colleagues, which is you know, is a funny start to a new role, but it's working fine. You know, we've been able to get laptops to people, and they've been, you know, plugged into our network, and all the introductions have been done over video, and it's worked fine.
And the Nasdaq listing, you kind of gone through the benefits of being there with your peer group, but I was wondering, are there anything else to consider? Are there particular costs? Is it very expensive having a Nasdaq and maintaining that listing? And then are there any other kind of reporting requirements, quarterly, or is it in the same schedule that you're currently working to?
Yeah, so we'll be what's called a foreign private issuer in the first instance, and an emerging growth company, you know, to kind of jargony terms, but effectively what that means is that we are exempt from many of the compliance requirements that a domestic issuer, a US company, would have. So over time, of course, we will have to report quarterly and have full, you know, Sarbanes-Oxley controls, but that is not an immediate requirement. So we'll build to that over the next few years. So for now, it'll be the same cadence as we currently have. full year and half year results, and then over time as we build out the organization, that will transform into more and more of a kind of US style company.
And the AstraZeneca deal, and you mentioned the timing of that and kind of it coming in at a time when from the outside you might have thought it'd be difficult to get deals done. Typically, I guess the sales cycle, how long does it take between starting conversation with a group like AstraZeneca or the other deals that you've done and something actually being kind of signed and sealed and delivered?
Yeah, so I mean the relation, typically these things take a long period of time in terms of that first introductory meeting and building the relationship. So that could take a year, or it's hard to put a precise number on how long that takes. But then really when there's kind of a meeting of minds and you decide that you're going to do a deal, the heavy lifting then is about 3 to 4 months or so. If both parties are fully engaged, there's a lot of work that goes on over that period. So we were deep in negotiations over January, February, March time, and really as the crisis kind of escalated up, there was meetings that would have been face to face and then moved to contract negotiations that would have been face to face moved to virtual. It didn't hinder it at all, and as I said, I think grateful and glad that AstraZeneca kind of saw the long view, and I think clearly the markets are kind of recovered to a good extent on the biotech side now at any rate, but it was still I think it was still what might appear to have been a brave decision in late March when everything was kind of crumbling around.
I've got a few questions around the GalNAc technology. I'll kind of put them all together if you don't mind. Kind of starting with kind of your protection, the IP around it, how secure that is, and then more kind of, I guess, in terms of its application, does the technology limit you to targets where you need to get into the liver, or can you develop? I'm going to read someone's words here, so forgive my pronunciation. Ligand conjugation to get the RNAi into other organs?
Yeah, no, it's a good question. So in terms of freedom to operate and intellectual property, we have a very strong position, as you would expect, and really, without kind of going into technicalities, I think the argument I would just simply make is, look, AstraZeneca, don't write an $80 million check unless they've clearly checked all of this out, and believe me, they did a lot of work on it. So that the answer is yes. And then you're right, GalNAc does limit you to the liver. You can only go to one particular cell type, and that they're called hepatocytes. Now, hepatocytes are probably one of the best cells you could think of if you're going to kind of sit down with a piece of paper and list your top cell types, because there's over 7,000 genes, proteins expressed there. So there's many diseases that you can target by going after hepatocytes, and it's not just liver disease. I mean, I've spoken about a cardiovascular disease program. That protein is made in the liver, but it does limit us. So we're trying to. There's a lot of value left in liver targets, but there's even more value in over the next many years in what we call extrahepatic. So targets outside of the liver, central nervous system, respiratory, cardio, oncology, etc. That's a big element of the AstraZeneca deal. So many of those targets will be extrahepatic, i.e. outside the liver. Mallinckrodt is liver targeted. Takeda is liver targeted. We are interested in further extrahepatic business development deals. We think that adds value adds optionality to us. We're more careful about liver targets because clearly we want to have a proprietary program, a proprietary pipeline that we can develop ourselves. But we are in conversations, in early stage conversations with some other parties around how can we use our technology to go outside the liver, and how do we do that in partnership.
And again, on the same sort of theme, how does the GalNAc technology differ to Ionis LICA or LICA technology?
Yeah, there's Well, so Ionis uses what's called antisense oligonucleotides, and they're single-stranded, and there are some biological differences between single-strand and double-strand. Ours are double-stranded. The body of evidence would say that siRNA is a more safe technology than antisense, but that's speaking in general terms. They use LICA, that's a very informed question, which is also a GalNAc conjugation, which allows them to also go to the liver. You know, it would be getting into to kind of extreme levels of technicalities to kind of precisely say that the differences. I think when you look down from the airplane, you know, they're kind of broadly similar technologies, but clearly there are nuances in how you attach it to the oligonucleotide and how you space it out and how you modify it, which gives each of us our respective IP around it. But you know, in broad brush terms, they're kind of doing the same thing.